They help in bringing the phosphate, which will be used during the transport. So sugars are transported this way, where they are phosphorylated as they are brought in. So two sugars are being shown, glucose being brought in, where when it comes in, phosphate from PEP across from a histidine containing protein, which we call HPR, brings the phosphate to the enzyme system. And this phosphate is then transported across from A to B. And eventually, as the glucose is being transported across, it will get phosphorylated. Mannitol is, of course, a sugar alcohol, and that also, when it is brought in, will be phosphorylated. Here is another model from another book where you can see mannose being brought in, and you can see that the not only are there A, B, and C components of this transport system, but there is a D component in the mannose. So they differ a little, but the whole idea is that when the sugar is coming in at that time, it gets phosphorylated. So why do you think the cell has to perform this maneuver right at the membrane? Uh, and why are the sugars phosphorylated? Phosphorylated sugars so important to us? Any ideas? So they're energized and they could enter into the glycolytic cycle very right away. Not only that, but once it's phosphorylated, it will not be allowed to leave the cell. So the reverse process is not there. So you're trapping the sugars inside the cell. Because sugars have different ways of entry, not only one type in one cell. So they may be diffusing out also by active transport or whichever system. So to phosphorylate it means now you put a tag on it and it is not going to escape from the cell once brought in. Another mechanism for transport is called the precursor product antiport. And this is, of course, an antiport system where instead of using ionic uh, sodium ion or a proton for exchange, here fermentation products are exchanged with the precursor. So you will see this in the lactic acid bacterium, lactobacillus, or even streptococcus, which utilizes the potential energy that is developed by the accumulation of the fermentation product. This potential energy will be used to drive the nutrient, the original substrate, into the cell. So let's look at it in a substrate product antiport system where different types of you will see either sugars or amino acids are being brought in by the buildup of the product inside the cell. So if you look at it, this is the periplasmic space, or the outside of the cell as it would be, and here is the cytoplasm part of it. So malate is being transported in, and the malate will be broken down by the fermentation, and malate will create lactate, and buildup of lactate will eventually drive out, and the malate will be brought in. So this is the antiport system, using the energy of the fermentation product that is created in here. Here's the arginine ornithine antiporter system, where the arginine is brought in, and through a couple of steps, it is converted to ornithine, and the buildup of ornithine will actually be the driving force for more arginine to come in. So this is again an antiporter system where the substrate is driven in by the expulsion of your product here. So these fermentation products are usually have to be driven out, which is not used. Therefore, this actually potential energy of that drives more of the substrate to come in. Here is your glucose galactose system also, which could be used as an example for the antiport system, where lactose is brought in, and using the beta galactosidase, when you break it down to glucose and galactose, the buildup of galactose will be the driving force for expulsion of galactose from the cell, and more lactose being brought in to go into glycolysis. So here's a summary of all different types of nutrient uptake systems in prokaryotes. And these are two models coming up from two different books, whichever you can look at. So if for your exam or your homework, if I give you a a system like this, transport systems, would you be able to express what they are if these words are not? Is what you should look at. This is your testing system. What is it? Is it a active transport, or is it a group translocation, or is it antiport, uniport, or symport systems? Very clearly, all the markings are there. Now, when you're looking at this figure, this passive diffusion of oxygen occurs both ways, right? Because this is easily transported across the membrane. Facilitated diffusion of glycerol also is both ways, you can see. But when it is active transport, all of these are active transports where energy is used by the cell. You will see that from the periplasm, things are going into the cytoplasm and they're not reverting back, except, of course, in your antiport system, your ions may be going in the reverse direction, or your substrate product exchange, where the product is the substrate is taken and the product is released out, right? So moving on, transport of we call it transport of iron, or iron uptake in the cell. So microorganisms also require iron, and this iron transport is very difficult for the cell because it's insoluble, and ferric iron especially is needed in the cell. So how do cells transport uh, a ferric iron inside the cell? They actually secrete what we call siderophores. Siderophores are compounds that help in or assist in the transport of iron. And two types of siderophores are the examples that I'm giving you. There may be more, but ferrochrome is one of the cyclic hydroxymate which is produced by fungi. And this ferrochrome, if you look at its structure, it is capable of binding a ferric iron, and this will help in transport the ferric inside the cell. Here is another one, which is an enterobactin. It's called an enterobactin. This is a cyclic catecholate, and this is produced by E. coli. And E. coli will throw out some enterobactin to get the ferric ion inside the cell across the membrane. Okay, so siderophores are known to form complexes with the ferric ion. And how are they transported? Once this ferrichrome or enterobactin has trapped the ferric ion with them, they will be using the transport systems, which we will call ABC transport system, right? Just like we just described, the ATP binding cassettes will be used to carry this molecule, the siderophore, with the iron inside the cell. So they don't enter just by themselves. They need transport system to be carried in again. Now, the reverse is what we will talk about. So we were bringing nutrients into the cell by different mechanisms, and I kind of briefly discussed those mechanisms. Now we're going to think about things that have to be transported from the cytoplasm into the membrane, either the cytoplasmic membrane or the outer membrane in case of Gram negative. And in some bacteria, these proteins may have to be secreted out of the cell also. So how does transport occur where proteins uh, have to be transported into the membrane, or in the periplasmic space, or in the outer membrane, or even outside of the cell? So we think about proteins as always having a three-dimensional structure. That means when they are created, they are a whole chain of amino acids, and then there is the secondary folding and the tertiary and the quaternary folding of the protein, and the protein becomes active once it has folded, right? So as such, uh, protein maturation occurs as a post-translational event, is what we look at it. That means once the amino acid chain is created, then it folds upon itself. So folding is required. Now, this folding of proteins is also associated with other proteins that may help either to deliver it to the subcellular or the periplasmic space, or these proteins may be used to excrete them outside of the cell. So definitely protein-protein interaction is what we'll be looking at, and protein maturation is what we look at. Maturation means when it becomes active by its three-dimensional foldings, then it will be a mature one. So protein folding requires molecular chaperones. We will call them chaperones. So these are molecules that are proteins. These are also proteins, and they aid in the folding of the nascent polypeptides. So these chains of polypeptides will be assisted by molecular chaperones to transport them. These molecular chaperones are not only needed for transport, but they also, in some cases, they protect them from thermal damage. Also means if you change your temperature from of a bacterium when you're growing it at 37, and then you take it to 42 degrees, and at that point, uh, this thermal damage can occur to a lot of proteins. So there are molecular chaperones that will protect those proteins that need protection from this increased temperature. So as such, if you look at it, a very good example is, we'll be talking about heat shock proteins later, but just giving you an example now, that when you shift a culture of E. coli from a 30 degrees to 42 degrees, and you very non-thinkingly, you would do this, taking it from one incubator to the other, within 5 minutes, the concentration of some 20 different proteins increased greatly. What is this for? What are these proteins coming up for? They actually are proteins that will protect the cell from thermal damage. So definitely the cell is very sensitive to temperatures, and heat shock proteins are very important in terms of protecting thermal damage to a lot of very essential proteins within the cell. So we will talk about heat shock proteins later, but to give you an example, the chaperones, molecular chaperones, are created or they are produced when the cell needs protection. These chaperones also aid in transport of proteins across the membrane. You will see quite a few of them as we go along as to how they help in the transport. So here is one example of a protein that are assisting in thermal, protecting from thermal inactivation. And here is your very important enzyme called E. coli RNA polymerase. What is E. coli RNA polymerase? Where is it used? Where would you find this polymerase? And what it would be function? Come on, please stop. What is a polymerase anyway?
For transcription.
One more time, please.
Sorry, for transcription.
For transcription, yeah. When the DNA has to be copied into an RNA, the RNA polymerase is the enzyme that is doing this copying of a DNA to a RNA molecule, right? Now, there are certain chaperone proteins that will protect this polymerase from a high temperature inactivation. DNA K is one of the proteins that is being shown here. The DNA K protects your E. coli RNA polymerase from thermal inactivation by, just a modeling of you to say that DNA K along with maybe DNA J and GRE participating and some ATP being used. Eventually, this unfolded protein will be protected and will be partially folded, and then the GRO-EL system will eventually allow the folded protein in its native conformation. So thermal damage usually unfolds the proteins, is what we are showing here. The unfolded protein has to be protected and then again delivered back to its original folded protein form where it will be active again. So just to show you chaperone proteins, and these mechanisms have been deciphered very well in some of these enzyme protection in E. coli cell. Another mechanism that will be operative on proteins once they are made is protein splicing. Now, you've been talking about gene splicing quite a bit when you do the genetics, but protein splicing also occurs to make a inactive protein or a pre-protein, as we call it, into an active protein. So, of course, here, removal of parts of the polypeptides will occur before the protein starts to fold. So these areas which are inteins, they are called inteins, and these are portions of the polypeptides that have to be removed. So if this is a whole protein, nascent protein that is being created, this is the N terminal of it, and this is the C terminal of it. And there is a intine in between, which a series of amino acids which are not needed in the complete protein. So the intine will have to be removed, uh, and then the two extines, N terminal and the C terminal, will be joined together, and your complete protein is created with the intine removed, just like your intron and exons in your genetic, in your genes, in your DNA. That splicing is occurring here. The protein splicing may be required for a protein to mature and to be able to use. So there are numerous protein secretion pathways that have been identified. They've been identified. Some of them will reside in all the three domains. Some of the protein secretion mechanisms will be found only in bacteria and archaea, but not in eukarya. But then there are some that are only unique to gram-negative cells. So why do we think that gram-negatives are always, in terms of secretion and uptake, gram-negatives are always mentioned separately from all bacteria. So what is so different about gram-negative bacteria? Simple question.
Cell wall.
Not cell wall. What is it in that cell wall that's important?
LPS?
Not periplasmic, but definitely the membrane, outer membrane. What is so unique about gram-negative bacteria? They have an outer membrane, which is again another membrane layer that will delimit what is going in and what is going out. Okay, so when we are talking about protein translocation and secretion in bacteria, you have to consider gram-negative bacteria separately because they have two membrane systems, the cytoplasmic membrane as well as the outer membrane. So remember, we are focusing on a membrane today. Periplasmic space is there, of course, that is important, but the membrane is the one. So this membrane also has a lot of proteins that will be the outer membrane. And these proteins are produced in the cytoplasm. They will have to be transported across the cytoplasmic membrane. They will have to be transported across the periplasmic space. They will have to be then embedded into the outer membrane. So you understand that's a very different mechanism, and a lot of different mechanisms will operate to transport any protein from inside cytoplasm of the cell to the either to the outer membrane or even outside. Some gram-negative bacteria do produce exoenzymes. Exoenzymes are enzymes that are liberated in outside of the cell. So some proteins that are produced inside the cell will have to go through the cytoplasmic membrane, will have to go through the periplasmic space, they will have to go through the outer membrane, and then they will be released. So there are different mechanisms of transport or secretion mechanisms. And this, because of gram-negatives having an outer membrane, it's of course, you have to study them very differently from your gram-positive bacteria or archaea. Okay, so there are two terms that you will come across. One is called translocation versus secretion. So word translocation capitalizes on the idea that proteins are transported across from the cytoplasm, from the cytoplasm where they are created or they're made. They will have to be going to the plasma membrane or the periplasmic space. Okay, so this is called translocation. Or even if it is going up to the outer outer membrane of gram-negative. And these proteins will include a lot of transport proteins, the electron transport chain proteins, proteins that are involved in chemotaxis. These are proteins that will be used in cell wall synthesis. There will be certain enzymes. So they will be brought from the cytoplasm to the plasma membrane or to the periplasmic space. Such a transport is called translocation. But if the protein is released in the external environment, we will call it a secretion system. And secretion will always allow these hydrolytic enzymes or the exoenzymes that will be used to break down nutrients on the outside of the cell. So please watch out for the term translocation versus the word secretion. So there are some common translocation and secretion systems. So we'll discuss each one of them separately. So the first one we're going to talk about is the SEC-dependent pathway. This is also called a GSP, or the general secretion pathway, GSP. it is dependent on a protein called SEC. So this is also sometimes called the SEC-dependent pathway. And this is the major pathway for all bacteria for transporting proteins across the plasma membrane. So this operates at the plasma membrane, and most of the proteins will use this pathway to get across the cytoplasmic membrane. Okay. Uh, this is this. There are others besides the GSP, which we will look at. One is called the ABC pathway. The other is called the TAT pathway. In gram negatives, since there's an outer membrane also, there are certain pathways of secretion in that. They may use sex system to bring the material up to them, sex system, which is the common GSP pathway, to cross across the cytoplasmic membrane. But they must cross the outer membrane or be embedded in the outer membrane. And those secretion pathways are only labeled as in Roman numerals as 1, 2, 3, 4, 5 systems. So these are operative only in the outer membrane, while these are operative maybe in the cytoplasmic membrane also, but they may be used in conjunction with these to allow the protein to get through the outer membrane also, or be embedded in the outer membrane. So whenever we are talking about secretion pathway 1, 2, 3, 4, 5, we are always talking about gram-negative bacteria. Have I made it clear?
Yes.
Wonderful. But realize that all these pathways, whether they are the operating at the cytoplasmic membrane level, or they're operating at the outer membrane level, they all require energy. So they're all energy dependent. The cell has to expend some energy to be able to transport or translocate those proteins. So we discuss each one of them, and the first one, of course, is the SEC-dependent pathway. This is also called the general secretion pathway. GSP is what we call it. This is highly conserved in all domains. That means your bacteria, archaea, and eukarya all have this general secretion pathway. It has been conserved in all domains. And it helps in translocation of proteins from the cytoplasm across or across the membrane or into the plasma membrane. So some proteins that have to be embedded into the plasma membrane use the GSP, or if it is to go across the membrane and go into the plasmic space, and this is also used now. Now, the condition for this proteins that are to be transported by this mechanism, they are always synthesized as pre-proteins. We will call them pre-proteins because this is not the folded stage yet, okay? So these are the condition to be transported across the SEC-dependent pathway. The proteins have to be made in a pre-protein form, and they have a leader peptide with them or ahead of them at the amino terminal. So at the amino terminal, there is a signal peptide, which we also call the leader peptide. Now, this signal peptide, which is in the pre-protein, what is its function? It delays the protein folding, because as soon as the protein is produced on the ribosomes, it starts to fold, right? But having a pre-protein leader peptide in the pre-protein will not allow it to fold right away. So there's a delay in the protein folding in these proteins which have the leader peptide in them. Then your chaperone protein SEC-B will be needed also to keep it in an unfolded protein. So there are two ways. The signal peptide is delaying the protein folding, and then the chaperone protein SEC-B will be needed to keep it unfolded and deliver it to the membrane. So looking at it in a picture form will be easier for you to look at this way. Here is your pre-protein, and this pre-protein has the amino terminal, which is being shown to have extruded out of the secretory system SEC-YEG. And here's your SEC-B, which is, of course, the one that is protecting it from folding again. And this is your SEC-A, which is, of course, an ATPase that allows the breakdown of ATP so that the protein can be transported across the membrane or be embedded in the membrane. So SEC-Y, E, and G, they make a channel in the membrane. You can see the sec-YEG making a channel in the membrane, whilst your sec-A translocates the pre-protein through the plasma membrane because it is an ATPase, so it will break down the ATP and allow the protein to be transported across. When the protein emerges from the plasma membrane on the other side, what will happen? There is an enzyme called signal peptidase